Well, first of all, thank you, you guys, the organizing, Adrian and Thomas and Zen and Larry. You have a really very useful approach, and we support it very much. It's also was quite amazing for me since the morning to be a little bit also frustrated, but underlines what you're trying to do, and that's to break the silos and bring them together. You know, we heard on the metabolic panel uh, and all they're concerned with is this diabetes, right? And we're asking about, well, how do we prevent not only diabetes but the rest of the aging? And I think that's a discussion that we're going to continue to have. Too much that we're not well informed between the branches. Also, thank you very much for the panel. Really, a dream team of geroscientists that are doing really incredible thing. And what we'll do is have several rounds where every one of the expert will give kind of an example of how excited and how progressive is our field. I mean, we went from hope to promise. We're trying to realize this promise. And basically the promise is that, well, aging is a biology, we all know because we see who's old and who's young, but that this biology drives age-related diseases, but it can be targeted, or then, as you said, preempted, and that health span and lifespan can be extended. So we will reveal now how to realize this promise, and I'll show just two slides in order to bring us, to bring not us, but the audience to the same level, sorry. So what we've done in the field, we kind of agreed that there are biological hallmarks of aging. The definition of biological hallmark of aging is they change with aging, and if we fix them by a variety of methods, we have an extension of health span and lifespan. That's how you become hallmarks of aging. In fact, Brian Kennedy is the first. You know, the United States came with those 7 hallmarks, and then European came with 9 hallmarks, and then there was Brexit. And so we're trying to organize those hallmarks. Those hallmarks are not necessarily mechanism, but they're very important to target. They're very important for the industry to pick up those targets and help us developing the Gero therapeutics. Let me just stop for a second and ask Brian, um, um, and sorry, I would introduce you. By the way, guys, read the intro. Those guys here have achieved so much, and I'm not going to spend the hour introducing only all those people. But Brian, who's now heading an aging center in Singapore, was the president of the Buck Institute, has been experienced anything from yeast to mice, and now works in humans. And he's one of the authors of the first paper of the hallmark of aging. Brian, just very quickly, what's missing here? Because this is work in progress. What would you put in the hallmarks or sub-hallmarks?
Yeah, thanks for the introduction and inviting me to join. Let me first clarify, we were the second paper, actually. The hallmarks paper came out first, and then we wrote the pillars paper. So we didn't, it was a culmination of a meeting as well. So there are many other people, including some in this room, that are on that paper. But thanks again, Nir. I think what I would say to that is, to me, it's those lines that connect the dots in the middle more than it is the words on the outside. Because whenever you look at an intervention, particularly in a mouse model, that extends lifespan and health span, it tends to affect all of the pillars or hallmarks. Maybe not directly, maybe that's a downstream effect of the primary function of the intervention. But what that tells me is that there's a network that connects these dots. And I think what's affecting longevity and health span are things that hit nodes that connect to multiple of these hallmarks. So the problem when I say that is that, you know, then we get into network biology and trying to understand how a system works that preserves health. And I think that hasn't been fully explained yet. So to me, it's really about understanding how things are connected rather than adding or arguing about one particular hallmark.
Right. That was going to be my second comment, but do you have, so for example, microbiome is not here. For instance, fibrosis is not here. Anyone want to jump in? Just that I want to move on, but not to tell you that this is a done story.
Yeah, I mean, I think you could put microbiome there for sure. I mean, we're looking at an intervention right now off ketoglutarate, and more data is telling us that it's affecting microbiome, and that may be a primary role by which it's targeting longevity. So we're looking at that very carefully, and I think you could make that argument for others.
Terrific. We'll go back to that. Now, we have Ana Maria Cuervo is here, who will cover proteostasis. Laura Niedernhofer will talk about genetic instability and maybe more on senescence. Vadim, and sorry, I forgot to say, Ana Maria Cuervo is the co-director of the Aging Center at Albert Einstein College of Medicine. Laura is the director of the Aging Center in University of Minnesota. Vadim is in Harvard, and he has this knowledge of how to take lots of data and use AI and use a lot of his experience and brain to make sense of everything. So we'll get to that. And Joan Mannick, I put her between epigenetics and metabolism. She's actually immunology, so she can cover inflammation and immune dysfunction. And she's converted to Geroscience, and she has both clinical experience and biological experience in aging. So that's what we're going to do, and that's going to be the base for our discussion. But I would separate this discussion this way also. If we look at pathway of longevity, we can think of 4 scenarios. One is life extension. The Stralberg's case is from Gulliver Travel. He came to one of those places where they had those Stralbergs that were immortal, but they continued aging. And it's a terrible story about them. And I don't think that's what we want. We don't want to have immortality with continued aging. So this is just for the framework. What we're going to discuss is, first of all, compression morbidity, the ability to live healthier for longer and be sick very short time at the end of their life. The Dorian Gray case, remember Dorian Gray stopped aging, but when he looked in the mirror, he saw his right age. By the way, try it. I'm doing that. I'm looking at the mirror and I'm saying, well, I myself am not aging. I'm just seeing that in the mirror. A really big task is the reversing aging, or the Wolverine case, or the fountain of youth, of taking old people and making them young. You know, that's very difficult, but we can do some of it, and maybe Laura will talk about it later. And then Joan and Vittorio will talk about Forever Young, really, about the Peter Pan case. You know, isn't it going to be easier just to take somebody at 20 and give them some therapy every month or every year and stop the aging altogether? I'll stop now the presentation so we can be face to face. And I'll ask Ana Maria, let's start with your hallmark. Let's start with proteostasis, and tell us, give example of what goes wrong and how do you fix it, and maybe pick up on what Brian says so beautifully, the interconnection between the hallmarks. Do you affect those others?
So thank you so much for inviting me to be here. And just for the general audience, I know proteostasis is an awful word, but basically just mean to make sure that in your cell, every protein is at the right time, in the right place, and at the right conformation. So if you think how many proteins you have in your cell, imagine how much work has to go into maintain them. But as you know, the proteostasis network has 3 major components, chaperones and 2 major proteolytic systems, proteasome and autophagy. And I'm just making this summary just to emphasize that wherever I say it's going to be on autophagy, that is the area that I know better. And we have heard already from Guido about that. But in a way, because of these interconnections, also there are very tight interconnections in between the proteostasis mechanisms. So whatever I say, it's going to apply to all of them. I just talk about autophagy because I know a little more. And I love that Brian started with the interconnections, because one of my big points has been to try to emphasize that when you think about autophagy, as you know, is just removing things from inside your cells, and in general for cleaning. That's the image that has been done, so you don't have damaged proteins. But autophagy goes much more than that. We hear from Guido how it remodels your mitochondria. It changes the number of mitochondria that you have. It also moves any energetic stores that you have. So it provides energy. It defends you against pathogens, has been linked to senescence. So you can find papers on this component of the proteostasis machinery involved in almost each of the pillars. And as I say, that's going to happen for everything that we talk today. And I completely agree that we don't know enough about these combinations. I mean, of course, in the case of proteostasis, my mom always say, in a clean house, everything works better. And that's the idea, right? You clean and things will work better. But we don't know how important that is, for example, for a neuron compared to a skin cell. And that's what we are learning these days. And of course, there are differences. And then we really have to integrate this at the level of organs. I don't think we can assume that the combinations that lead to a successful aging are always the same for every organ or for every genetic background. And I think that's where I see that we should move in the future, is trying to understand what are the right combinations. There's not going to be a perfect one, but there is going to be one that fits your particular characteristics.
But Anna Maria, when you target chaperone-mediated autophagy, which is one of your favorite, and you get an extension, a remarkable extension of lifespan, what other things happen to the body is related to the other hallmarks, maybe?
So that's great. I was not sure if we can talk about our own data, so I've been trying to avoid chaperone-mediated autophagy.
You can if I ask.
Yes. So we have prepared all this. So this is a very selective way of removing proteins from inside the cell, and something very important, not only because there are damage, you can remove fully functional proteins to terminate their function. So it has very important regulatory functions. And what we have found is that when you prevent the decline with age, notice that I don't say when you enhance crazy when you are young. No. You just maintain the same CMA activity that when you were young, but in an old animal. The animals live longer, and it applies to both genders. With Rafa de Cabo, we have been doing all this frailty. They are less frail. They have lower amount of senescent cells. They have beautiful metabolic energy expenditure compared to the old ones. Also, they recover circadian rates. That is something I think we have to start thinking to include in this chart, like the circadian and how important it is. They have better inflammatory response that the group of Fernando Macian has shown. Their stem cells, we published very early on this year that the activation of stem cells in all animals that have preserved CMA is better. So basically less DNA damage. So almost for every pillar or for every hallmark of aging, you have a beneficial effect. As much as I would love to say, oh, it's all about CMA, no. I mean, I know that some of the effects are because your mitochondria are better or your DNA repair mechanism is better, but our only intervention has been modifying CMA. So this kind of reinforce these connections, and that you have much better effect than what you predict by acting in a single pathway. Even your intervention is still in a single pathway.
Right. I always say to more in a more simple for more simple explanation that if you take the cell or the organ or the body and make it younger, you fix a lot of things by that, and then it's hard to know what's primary and what's secondary. So we're still at the Dorian Gray part, and Brian, back to you. Can you give two examples? You started with one of Gero therapeutics, because we geroscientists want to develop Gero therapeutics or Gero protectors that can extend health span and lifespan. What's going on now in your lab or in your world?
Yeah, I can talk a little bit about that. I think first of all, there's probably 20 to 50 of these geroprotectors, maybe more, that have been proposed, and there's varying degrees of data on them. I think one of the exciting things we're doing in the lab is to begin to test them side by side in human clinical studies, and so hopefully we can start to tease out which ones work in which people for what reason. I think I mentioned alpha-ketoglutarate, and that the reason I like that so much is it's a safe, natural product that compresses morbidity in mice. So it extends lifespan by a little bit, but reduces frailty by a lot. We have some human clinical data that we're getting ready to publish on that, although I must say it's not clinically controlled. We also have a placebo-controlled study that we're still completing. But there's at least indications that things might be interesting in humans as well. And then we're looking at a lot of different interventions in animal models, and have still not published this, but we've been looking at this fibrate called gemfibrozil that is used for hypertriglyceridemia, and it has some very interesting properties. And we think what it's doing is inhibiting amino acid transport in the gut. So we think it has an entirely new function that's not been identified before. It's thought to be a PPAR alpha activator. So we're putting a lot of time and effort into trying to understand if it really does affect amino acid import, and if that's why it's affecting longevity and health span.
Terrific. So I want to move now to the Wolverine, the reversing aging thing. And I know, Laura, you already in the last session said we're going to extend health, but not lifespan. So it's not exactly the fountain of youth, but tell us some of the examples from your lab, from other labs, of how do you show that senolytics is so center in treating aging?
I guess the way I'm thinking about it is the way Anna Maria is talking about, like, the garbage inside of a cell. I'm starting to think of senescent cells as they've hit a point of no return, whether you really don't want to reverse senescence because that's a potent tumor suppressor mechanism, but those cells are just damaging and pro-inflammatory and virtually have been linked to every age-related disease. So if you could get rid of those, just like you could get rid of your damaged proteins or repair your DNA, I think you're going to be in a lot better shape. And so right now, senolytics, these drugs to clear senescent cells and to dampen inflammation, they've been tested in a wide variety of animal models. And I think it's particularly exciting to think that it might have some benefit in Alzheimer's disease, where we desperately need new therapies. And then I was also really excited to see it worked in an animal model of beta coronavirus infection, and the whole idea that rapalogs or senolytics could affect this situation, modeling COVID-19 infection, really speaks strongly to the geroscience hypothesis. I mean, here's an individual, an elderly individual who's just particularly vulnerable to the bad outcomes of a new infection. And I think that speaks volumes about the fact that there's a biology of aging that just needs to be targeted.
Terrific. Vadim, to you, there's a lot. What we've done right in our field, I guess, is we picked up models that live much longer, and we learn a lot about the biology. And it shortcut a little bit our way to gerotherapeutics, but aging is really complex. And you're really incredible in the sense that you take lots of data and find a needle in a haystack without increasing the haystack, which is what most people do. Tell us a little bit of your interest in the gerotherapeutic aspects of aging.
Yeah, thank you, Nir. I would answer a little bit broader, I guess, just to introduce also the clocks, the epigenetic clocks, because over the years, there have been many ways developed to to follow the aging process. You could, for example, look at the telomere length or oxidative damage, or I don't know, mutations in mitochondria, or there are so many parameters, but it is the integrative biomarkers like epigenetic clocks, which I think changed everything in the field. So the clocks were conceived by Steve Horvath, and they allow to quantify the aging process. And suddenly we ask questions which I think may be just central in the field. Of course, clocks they are different. So, some clocks may be trained for biological age or chronological age or future mortality or phenotypic aging and so on. And this kind of reflects the disagreement in the field because there's no consensus as to what aging is, because some of us think it's increased mortality and other people think it's decreased function or increased biological age or increased molecular damage. So all of these features represent the aging process. In the second half of our lives, actually, they all apply. However, when we try to target the aging process, we could target various different aspects of aging. So whether it's mortality or biological age or damage, molecular damage, and so on. And that's the beauty of the clocks, because clocks can be trained on all of these features. Of course, they are still in development, and various clocks that quantify various features of what various people think aging is, they're still in development, but they already exist, and therefore they could be applied in many situations. And that's what we try in the lab as well. We try to develop epigenetic clocks. I would just comment just very briefly that we developed a single cell epigenetic clock. The paper is currently in press, and so basically it allows to quantify aging at the level of individual cells with from a very few sequence reads, about 10,000 reads, and kind of related to that, one could quantify aging in bulk samples also from very few reads, sequence reads, like 10,000 reads, which allows to decrease the cost of estimating age from epigenetic clocks maybe 100-fold to the level of less than $1 per sample. So we're very excited about this development, and hopefully we'll see how it develops in the coming years. As to interventions,
so let's wait. You switched my questions. I'll get to intervention later. Just a follow-up question. Look, the clocks are important mainly to know your biological age versus your chronological age, but they have to serve a bigger purpose, and that's when we try those Gero therapeutics. Within weeks or months, we know that they are working on aging. So how do you plan to look at the biomarkers that are relevant for testing of Gero therapeutics?
I think that any clinical trial or any large scale study in the aging field involving, for example, human samples or genetic screens and so on, they must use epigenetic clocks or maybe some other biomarkers if they're developed and which are better. But this tool really, in my mind, changes everything. It's a revolution in the field. And I don't know, maybe some people have not realized that, but to me, this is true.
Terrific. And now I want to go to the forever young thing. And Vittorio, I realize I haven't introduced you, but you're in Stanford. Thanks for waking up for us. You've done an incredible job on stem cells. So tell us, I think what really was important for us was to know that you can take a sperm of a 70-year-old man and an egg of a 50-year-old woman, and we can measure, Vadim can measure their age probably, but when the blastocyst is formed, we erase the age of the parents. So we have this built-in system where we can erase the age. So how do we erase the age later on, Vittorio?
Yeah, hi Nir, and hi everybody. Well, that's a great question, and frankly, this has been the inspiration of our work over the course of the last 10 to 15 years. I have to say, which is exactly what you just said, like realizing that there is already for an evolutionary kind of goal, there is already a machine that exists in nature that is responsible, for erasing. So the machine was built for other purposes, right? To, as you said, to change the identity of two fully differentiated cell types, the sperm and the egg, and make a new organism out of those. But embedded in that molecular mechanism, there is already a lot to learn about how epigenetic marks, for example, of aging can be erased, reset, and wiped out. And this, in my opinion, this has been the inspiration for two fields which will have a lot to say and to do in the future, for geroscience and in the longevity field. The first, of course, is the discovery of iPSCs, because the work on nuclear transfer was the inspiration somehow for the work that led then to the discovery of Shinya Yamanaka of iPSCs. Which is the kind of the simplification of what nature does with reproduction in a test tube by reprogramming the somatic cells to iPSCs. And you can think about iPSCs, and we have endless examples about that you can really use iPSCs to generate in a cell autologous fashion, in an individual fashion, unlimited numbers of stem cells, tissues, organs. Of course, the field is developing, but there's endless examples of how iPS technology can be utilized to generate new organs, new tissues, which are youthful, and they can replace a dysfunctional aged organ, in any patient virtually. And the second field that I think is now taking off is the idea that if you control that reprogramming process without going all the way back to an undifferentiated state, which is the iPS state, if you can control that reprogramming process very tightly, very, very finely and precisely, you can actually, without going all the way back to iPSCs, you can rejuvenate, bring the clock back virtually in any tissue, any cell type in the body. And maybe not in the so near future, but very soon, I think that we will be able to really start thinking about therapies that are going to be able to reverse the clock, whatever that clock is, reverse the clock of any cell type. So, and again, you can think about this going back to the Peter Pan kind of analogy. You can think about applying this to aged individuals, so you can reverse that process, or you can prevent the process from happening. And so, exciting things to happen in the future.
Terrific. And Joan, you've been working with Life Biosciences, and they have a project of rejuvenating an organ. Can you all publish? So I'm assuming that there's no secrets that you're going to share, but can you talk about this concept of rejuvenating an organ, and how do we go from here?
Absolutely. But if I can, I want to brag about the work being done by two of the panelists, since it's not my work. And then I want to talk about this very exciting part of life. Laura has published a paper this year in Nature showing that aging of the immune system drives aging of every other organ system. This is a landmark paper because the immune system is a dynamic organ system that's malleable. So if we can target the aging immune system to prevent aging of other organ systems, that's a huge sort of exciting area of medicine. So I want to shout out to Laura for that work. And then Anna Maria has published an amazing paper in Cell this year. What she didn't talk about is this CMA, which she activates with compounds she's developed, and we have actually at Life. This process is degrading the proteins involved in a lot of neurodegenerative diseases like alpha-synuclein, tau, Huntington. And part of the reason neurodegenerative diseases may develop is because of declines in this activity, the CMA activity. And Anna Maria published this paper showing that if she activates with small molecules this CMA activity, she has really dramatic benefits in frontotemporal dementia and Alzheimer's disease models. That's a whole new area for neurodegenerative diseases, a whole new target. So, I mean, amazing work for Anna Maria. And then what you asked me about, Nir, was studies that were done by David Sinclair and also published in Nature last December that are kind of remarkable. And as Vittorio was saying, Yamanaka factors, they're 4 proteins that can actually turn an adult cell back to an embryonic cell, sort of erase age and go from old to young. The problem is when you use 4 Yamanaka factors, those embryonic cells when you do it in vivo, turn into tumors called teratomas. So it wasn't clear, can you reverse age safely? And what David did is, instead of using 4 Yamanaka factors, he used 3 and removed one of the Yamanaka factors that are associated with tumor development called c-Myc. When he took a gene therapy that induced expression of just 3 Yamanaka factors and put it in an eye, he could actually restore vision that had been lost in a mouse model of glaucoma. And also he could regenerate an optic nerve, which normally can't regenerate in adult animals. So as Vittorio was saying, this opens up a whole therapeutic area where we might actually be able to not just stop aging, but reverse aging of organ systems. And that's, remarkable. And we just have to carefully move this into the clinic and make sure we do it safely.
Terrific, and thanks for mentioning that. And you see, Anna Maria and Laura, if you don't break, somebody will break for you. And I totally agree. Those were amazing stories. By the way, the only thing that in association trial, not clinical trial, restored the immune system and also the ability to sustain a severe disease is metformin. 9 papers around the world show that metformin has less hospitalization in COVID patient and 1/3 of the mortality. And it's, again, all geroscience, all this connectivity between the hallmarks. Okay, so I want to go now to the second round, and some of you were enthusiastic enough, or I wasn't holding you back, and so we're going to up our game even more, and talk about what's coming next. Bill Gates had this saying that we're overestimating what we can do in 2 years, but underestimating what we can do in 10 years. So let's take 10 years and let's see what we can do in the next 10 years. Vadim, I want to start with you. I actually was thinking of the biological clocks here, but can we just regress for a second and tell us about the way you're thinking of developing gerotherapeutics?
Sorry, Benir, I'm a little bit confused by the question. So whether you're asking about the kind of development in the near future, or it's like you said, 10 years?
Well, what you expect in 10 years, but I want to start by your algorithm of how do you find and test geroprotectors?
Okay, so I will answer, I guess, both kind of aspects of that question. So we are developing kind of quantitative approaches, unbiased approaches to identifying interventions that extend lifespan. And actually, it turns out that the interventions that we predicted based on so-called longevity signatures, there's no time to go, to the specifics, but basically we have like computational patterns of gene expression, and we are able to predict compounds, and actually they indeed extend lifespan. So we are very excited about that, because this would already adding quite a few compounds in mice that extend lifespan. But I guess the next question is how to translate this to humans. So currently, and Brian already mentioned that we have already many compounds that work in mice, and many of them in principle should work in humans. It's just they need to be tested. So instead of, I think, just metformin, as of course it's a great idea to test metformin through a tame trial, but I would suggest that we would need to test many compounds in humans, and some of them would work, and hopefully this would really change the field once a safe compound can be found that robustly extends lifespan, and there's no reason why it shouldn't work. So this might change the field, and it could benefit the field in many different ways.
So interestingly, biotech's investments have gone down in the last year, but not for longevity. The biotechs in longevity went from $1 billion investment to $4.5 billion investment. So the investment is ahead of how others are seeing gerotherapeutics and how the FDA is looking at gerotherapeutics. Laura, I know that you've been following Geroscience clinical trials. Can you give example of three examples of what do you think is coming in the next 10 years?
Well, I think because the clinical trials with senolytics and NAD precursors and others have been initiated, and they've been very thoughtful. The field has been working together to think about how to advance these safely and stepwise. I think we're going to learn about target engagement and safety, which are the first priorities, as well as beginning to see if they actually work. I suspect, and I've heard Ned Sharpless even say this, that within 5 years, we should know if any of these geroscience platforms are really going to have any efficacy. And then designing the trial where we're really talking about treating aging as opposed to a disease endpoint is still going to take some work. But again, I'm encouraged about some of the short-term studies. Hopefully COVID-19, that would be great. We could use another therapy there, as well as thinking about new approaches for Alzheimer's are kind of what I'm most excited about.
Terrific. And Joan, you are in the midst of trials with the FDA on Gero therapeutics. So first of all, if you can talk a little bit about Rapamycin and what did you learn from this process of bringing a new field out there and waving and saying, hey, here we are?
Yeah, it's a great question, dear. So when we were developing mTOR inhibitors to improve immune function, and this was a new endpoint for the FDA, and they said to us, we're going to have to learn together how to develop this kind of a drug. And it is a learning curve. So our first shot at this, I don't think we got the endpoint right. You know, it was an endpoint the FDA thought would be a good one, but it turned out probably isn't the right endpoint for an mTOR inhibitor. But I think we have to be willing to say, as we move into these new areas, we're not always going to get it right, but we're going to learn. And the more we learn, the more we're going to get it right in the future. And so I think we have to have patience and sort of perseverance. And I agree with Laura. In the next 5 years, one or more of these interventions, we're going to get the right trial design, the right intervention, and really move the needle for medical care.
So the geriatrician are saying if somebody comes to them and say, you know, we have pain here and pain there, they say, if you wake up in the morning and you have no pain, you're dead. Okay, and the point here is the elderly are complaining, and yet the FDA wanted you to have subjective measurements of aging. That hasn't been proven to work before, right? That's one of the lessons to learn, isn't it?
Yes, so the FDA for a phase 3 endpoint wanted an endpoint that reflects how people feel and function. So for a phase 2B trial of something improving immune function, they wanted laboratory confirmed infections. For the phase 3, they said, we just want symptoms. We just want to show that you could decrease the symptoms of infection. But as Nir said, what we learned is in the elderly, they have a lot of symptoms, and it's really hard to tell symptoms from infection from symptoms due to other things. So it was too vague an endpoint. So I think we're learning we need more targeted endpoint and more targeted patient population, and I suspect in future trials we'll be able to hit.
Terrific. Anna Maria, maybe you could make some comments about, you know, you working with genetics tool to show what's the impact of your discoveries, and going into developing a drug. What do you think are your major lessons? What would you tell other people who are looking at this possibility?
Never do it again. No, I'm just kidding. No, I think that's a very good point, because I feel that each one should do whatever their expertise is on. And I feel like, you know, as academic, the part of discovery, I think we are good at. If we will have not partnership with experts like Joan that know what they are doing, I think we will be very behind. And that's not on benefit of anybody. So I think the best messages or the best things that I learned is to really start having great collaborators. I mean, I got an amazing chemist, Ebris Gabaciotis, working with me from the beginning. And we learned through a lot of errors on my part. And then as soon as we could, we really team up with the industry, because they are the ones who can really push this forward. And I think my time and my little knowledge is much better used when I focus on the basic. And of course, I'm learning a lot. We are heavily involved. We are lucky to participate every week in a discussion with them. And I'm learning all the things that we did wrong, but I think this is the way to go. And, you know, unfortunately, we have different points of view. So the outcomes that we expect are very different. And I think that's something that took me time to understand, that once you move into real people, this is a change, a completely change of game here. But I still think for anybody interested in doing this, that is extremely important. And I also notice a big change as I interacting more and more with industry on how much they appreciate the basic science. And I think that's very important. I mean, for every young person listening to us, if your heart is into the mechanistic part, still follow that. Not all of us, I mean, I don't think I'm gonna cure anything. Other people will do that or anything, but experiments and studies coming from our basic science labs are being fundamental for people who know what they are doing to take them and develop them in interventions. So, you know, big defensor of the basic science. But if you have the opportunity to team up with experts, I think it's a great learning experience.
Yeah, I totally agree with both points. You know, mechanism is crucial, and with the fact that biotech, you know, is a different profession. Okay, don't think that your PhD studied something in groups of 8 mice and you know to develop the drug. Both are true. Vittorio, what do you think will happen in the next 10 years in your field?
Oh, a lot. Probably in the next I should say the next 5 years. No, as I said before, I mean, there is already a lot of efforts done both at the academic level, but, you know, at the biotech level as well, on the generation of patient-specific organs or tissues that can be used, you know, for a variety of different conditions. So this is already happening, and in the near future, absolutely, there's going to be a revolution, I think, thanks also to the development of the technology, of course. AI, you know, plays a central role in this as well, in particular when it comes to, you know, the differentiation of the cells to make the process very efficient, reproducible, and so there's going to be a huge development, you know, on the generation of patient-specific tissues and cells, but also on the idea of, as I said before, on the idea of transient reprogramming or partial reprogramming or controlled reprogramming. I mean, there's many different ways to think about that. There's going to be also a lot of developments. There's already different biotechs that are working on this, Life Bio, Turn, my company, and others. So things will be moving fast, and of course, this is going to be a synergy. It's not going to be the solution, you know, or the solutions. You know, this is going to be a synergy across many different interventions that all together, I think, are going to have a profound impact on longevity.
And Brian, you in Singapore, you're 12 hours ahead of us, so you see the future much closer. So what do you anticipate?
You know, I think that there's two things I want to bring up. One is that, you know, I was talking to Morgan Levine on our aging seminar series. We have this for free, by the way. Anybody wants to go to it, they can just go to NUS Healthy Longevity webinar, and you can find Mir or Joan, and the rest of you, I know where you live, you're going to be on too, talking about aging. Anyway, I had Morgan Levine on, and she was trying to sort of go in reverse from the methylation clocks. So to take all of the different methylation sites that have been identified in different clocks and work backwards to see which ones are linked to which hallmarks of aging. And I think that's a great way, and I'm sure Vadim thinks about this too and could comment maybe. I think that's a great way to try to understand the relative contribution of different hallmarks in specific individuals. So this might be a nice path to get to personalized aging, and then ultimately might link to different interventions that could target different pathways in specific individuals to have the biggest impact. And just to add one more thing on interventions really quickly, I think that it's going to be really important to start looking at combinations of interventions, because a lot of people out there, particularly with supplements, are taking 5 and 6 and 10 things right now. And we've done a fair amount of stuff with 2 and 3 things in a mouse. And let me tell you right now, it doesn't go like you might expect. It's not always additive or synergistic. Sometimes 2 things antagonize each other. And I can't really even predict what's going to happen before the experiment starts. So I think the concept of hitting multiple pathways to get bigger effects is a good concept, but that's not going to be as simple as we all, well, maybe we don't think that, but that's not going to be as simple as some people think. And we have to really start looking into that, even in preclinical models, to get some idea where to go, because I wouldn't want to see a lot of human clinical studies where we're having 2 molecules that antagonize each other.
Right. And back to the personalized medicine and trying to look at the hallmarks apropos the methylation, you know, we looked at proteomic 5,000 protein in 1,000 people between the ages 65 and 95, and we asked what is changing. And one of the incredible things, a lot of what happened was breakdown, you know, breakdown of collagen or degranulation of cells, extracellular metrics. And at first we thought it's a noise, but then I thought, you know, however we target aging, maybe that's at the end what we have to see. Maybe it's the best biomarkers. So there could be more public biomarkers and more specific biomarkers to target the therapy potentially. So we have 10 minutes to go. So now I'm going to open it and ask each one of you, what did I miss? What do you think I should have asked you that is really important? You know, about a minute for each one of you. So let's go on my screen. The first is Vittorio.
Oh man, this is a tough one. What did you miss? That's a great question.
Do you want to disagree? Do you want to disagree with somebody that we said? Did we forget anything important?
No, I just wanted to follow up on what Brian just said. You know, he is absolutely right. I think that those studies need to be done because some pathways may work in an antagonizing fashion, but some actually may work in a way that they're not additive, but to some extent even exponential, in the sense that, you know, in some cases we may see that if we trigger the right or if we touch the right chords, we may get really a symphony out of it and not cacophony out of it. So I think it's absolutely important to first of all understand. There's still a lot to understand, you know, at the level of what aging means at the molecular level. We all know it happens, but we still have a poor understanding of this process. So we need to understand that better, and then understand how, you know, we can touch the different pathways and make them work together in a synergistic fashion.
Terrific. Joan, maybe I can target more. How do we face the FDA that don't necessarily believe that aging can be targeted? What are some of the approaches we should consider?
I actually would push. I think the FDA is open to anything where the benefit outweighs the risk. So I don't actually think the FDA is our issue, but I'm seeing all these questions in the chat room about aging as an indication. And I think this gets to the importance of the TAME trial. So what differentiates aging? Aging biology, it's a risk factor for multiple different diseases. So targeting aging is different than targeting other biologic process because it could benefit multiple diseases at once. And how do you show that in a clinical trial? So this is Nir's work. This isn't me again. He had this really creative idea of saying, why don't we take people who have one or more aging-related diseases or low Viking speed, give them a geroprotector, in this case metformin, and see, can we delay the onset of another aging-related condition? So this is a really interesting kind of multi-organ system aging endpoint that I think, if it hits, is a sort of a roadmap for how we could move other geroprotectors through on this sort of more broad multi-organ system benefit endpoint. And Nir, I'm going to pass it to you because you're the expert.
Well, I'll just say, and when we talked with the FDA, we realized that we don't want to call aging a disease. By the way, elderly don't want to be called sick when they're 65. Okay, especially after the pandemic. ARP doesn't want it. FDA doesn't want it. The government doesn't want it. We don't need to. We said, our outcome is a cluster. We're moving a cluster of age-related diseases, and everybody can call it what it is. We know what it is, and they can think that it happens that metformin does 5 different things by accident, unrelated. God created this wonderful thing. Okay, Anna Maria. Final thoughts.
For the future, I have aspirations at different levels as a community. I think that the most general is that we should take, and I think when we put together all these hallmarks of aging, we just talk of this as a working frame. So nobody should think that these are written in a stone, and I think this is very important. And the idea was test and refute, as we've been doing during this year. So there is room for many more things, and I hope we will learn many more about those ones. From the point of view of biology, and in the case of proteostasis, I think we know a lot inside the cell, and it's time to understand a bit better interorgan interplay on this. We don't age in all the organs at the same time. There are some vulnerabilities, and I think we should exploit on that and how proteostasis in each of them change. And then from the point of view of more apply, I think for biomarkers, and we haven't talked much about that, but for all this, we really need to know the baseline. We don't even know each of these hallmarks of aging. What is normal? We haven't talked about diversity. I mean, population is going to be very different, and we absolutely need that data to come and build awareness, biological questions. And then from the point of therapeutics, I think knowing the timing, and summarizing a little what has been said, I mean, these combinations, they might not be so good when you do it late in life, because one of the changes might be reactive. So you might not want to fix down your autophagy if it's just reacting to the lack of energy in the cell. And I think this timing is going to be very important. So those are my 4 points.
Laura.
Well, just to amplify what Anna Maria is saying too, I think any efforts that we can as a community to break down silos is going to be really important, so that all the different geroprotectors are tested in different disease models, and that you have help measuring each of the hallmarks of aging very well, because we tend to be experts in 1 or 2 at the most. The other thing I'd like to make a plug for is, I think where the geroscience field can have a huge impact is improving animal models of diseases. And so I'm very optimistic right now, because I'm seeing a lot of people suddenly interested in considering age as a variable in their cancer model or in their neurodegeneration model. And this is going to be really important. But the thing that spooks me a little bit is, I think we're neglecting the environment. And when we go to translate to humans, that's going to be a big issue. So we do need to think about microbiome and not being SPF all the time. So I think that's our gift to the community.
And I wanted to mention about those hallmarks, that exercise and diets are affecting all those hallmarks. So the environment is clearly important. Vadim, we have a couple of minutes more.
Yeah, I would like to actually highlight the area of rejuvenation. I mean, not to say that geroprotectors are not important, they're very important, and I agree with what has been said. And on rejuvenation, Vittorio and Joan, of course, mentioned, but I would stress that this is for the first time we know that certain cells, maybe not entire organisms, but they can be rejuvenated, whether you apply reprogramming, so for example, partial reprogramming, or well, we actually published a paper a few months ago when we found that certain cells at certain stage in embryogenesis are rejuvenated naturally. This is the first example of natural rejuvenation. So there are other examples. Basically, in principle, it's possible to reduce the age, biological age of certain cells and certain organ systems in a body. What does it mean conceptually when like certain cells are younger than other cells, so certain tissues are younger than other cells? How to affect aging, and actually what aging is in this context? All of these are open and very exciting questions.
Very, very good point. And Brian.
Let me unmute. First of all, I want to echo Laura's point about age-appropriate animal models. You know, one of the things we did in US is set up a budget. So now if somebody has a reason to have aged mice, we just buy it for them, because I've been saying this for a long time, and I think that that's a real issue with disease models in mice. But I think what I'll take 30 seconds on is to talk about the dog aging project with Daniel Promislo and Matt Kaberlein, because I think this is a great interface between preclinical models like mice and humans. You have aged dogs living in an environment with their owners, and this study is a really nice design to look at the diversity of aging within a canine population, which is quite a diverse genetics, and also to look at interventions. And so there are intervention with rapamycin ongoing now, and there are others planned maybe to be tested. So I think these companion animals may be a great place to really convince people that something can be done about aging and get some data that correlates better with human aging.
Terrific. And I would like to say as the end that this is not only about aging. When we treat cancer with radiation and chemotherapy, we age the patient rapidly. Kids are getting heart disease at age 30. So we need to help those people. HIV, people with HIV get diseases 10 years earlier than others. Disabled people. And if we want to go to Mars, we have to stop aging, because we'll get there with disease and we'll never come back. I want to thank you for really a great discussion. I think we really affected the people who listen to us. And thanks, Thomas, and you guys, Adrian, for allowing this to happen. Thank you.